Hi, Dr. Biology here, and this section is about the control of gene expression, and it's 3.8, part of the AQA specification for A-level biology. So first of all, we're going to look at gene mutations. So mutations can be caused by errors during DNA replication. So this is very much related to the work that you might have done before on protein synthesis. Changes of exons in the bases can result in a change of the sequence of amino acids, and therefore this might result in a protein becoming non-functional. So we need to understand the different types of gene mutations. So in year 12 you probably did cover this section of work looking at three specific gene mutations, but there's a few more you need to know about. So firstly, let's talk about substitution. So substitution, as you can see in this diagram, is where one or more bases are swapped for another. So in this diagram you can see G has been replaced by A, and that is therefore the triplet code, or the codon is rather than glutamine, it's now coding for lysine. So this sometimes can result in no changes, an amino acid sequence could stay the same due to the fact that the DNA codes, triplet codes, have degenerate nature of the code, where amino acids are coded for more than one DNA triplet. Deletion. So in this example, deletion, you can see that A has been deleted, adenine, and therefore it's causing what we call a frame shift. So the triplet code is read differently and it changes all the amino acid order from that deletion. Addition is very similar, so it's where there's an addition of a base. This again may result in a frame shift the other way. So the triplet code is read differently, changing the amino acid order. Duplication. Duplication is an interesting one where one or more bases can be repeated. So in this case you can see the triplet code TAA is repeated, and it can be repeated quite a few times, not just once. And again, it causes a frame shift, but it can also result in increasing repeating subunits and causing quite serious genetic disorders. Inversions, so it's a sequence of bases that are reversed. That can result in no change, as the amino acid sequence could stay the same, and it's known as what we call a silent mutation, where it has no effect. And then translocation, a sequence of bases is moved from one location in the genome to another. So this could be movement within the same chromosome or to a different chromosome, and this is going to have a major effect on protein synthesis. So just to summarize, not all mutations affect the order of amino acids. So for example, silent mutations, this is because amino acids are coded for by more than one DNA triplet, and that's mainly in substitution and inversion mutations. Whereas more serious are frame shift mutations related to deletion, insertion and duplication. So these have a huge effect on the base sequence, changes the number of bases in the DNA code, causes a shift of all the base triplets so that all codons after the mutation are changed, and the DNA code will be transcribed differently, more than likely result in a non-functional protein. So let's move on to the next section related to gene expression, which is stem cells. So you would have come across stem cells before, and probably you would have come across them back in GCSE. So let's have a look at the different types of stem cells. So first of all, stem cells are unspecialized cells that can develop into other types of cells, and you need to know the four main types. So the first is totipotent cells. So they're only present in mammals in the first few cell divisions of an embryo, and they can specialize into any type of cell. Second one is pluripotent cells, so after the embryo develops, specializing again into any type of cell except the placenta. And then multipotent cells, they can differentiate into related families of cells. So it's things like blood cells, muscle cells, skin cells, and these are known as adult stem cells. They're used by the body to replace and repair damaged tissue. And then finally, unipotent cells, they can only differentiate into one type of cell. So for example, cardiomyocytes, which are heart muscle cells. Now, in terms of use of stem cells in medicine and research, obviously the use of embryonic stem cells has caused some issues, some ethical issues, which I'll talk about in a minute. So one of the ways that researchers have overcome this is using what we call induced pluripotent stem cells, or IPS cells. So that IPS cells are unipotent, so they're taken from an adult, okay, so an adult stem cell, and they get genetically reprogrammed. So what they do is they add certain genes to the cell, and therefore they make it act like it is a pluripotent stem cell. This is a new development. It's still at the research stage, but they may solve some of the problems of using embryonic or embryo stem cells. In the AQA spec, it does talk about the fact that you need to be able to evaluate the use of stem cells in treating human disorders. Now, more than likely, they will give you background information or application information in the stem of the question. However, here are some kind of, quite common pros and cons, because remember, when you talk about evaluate, that's looking at the advantages and disadvantages, or the pros and cons. So the pros of using stem cells is that they can be used to grow new organs and tissues for treatment of a variety of conditions. So in this case, I've got Parkinson's disease or spinal cord injuries. It is also possible to use adult stem cells for some treatments, which people feel is more ethical. However, they cannot develop into all the specialized cell types. So I suppose that's a con of using adult stem cells. And you can use stem cells to replace diseased or damaged organs, such as the bladder, or to replace damaged tissue, for example, in the trachea. Now, the main con in using embryonic stem cells, and they are from unused IVF embryos. Some people object to this, as they see it as a destruction of a potential fetus. So some people will believe that as fertilization has happened, the embryo has the right to life. So in terms of expression of genes, and therefore expression of proteins, we need to talk next about the regulation of both transcription and translation during protein synthesis. So I'm going to give you a reminder, first of all, about protein synthesis, something you would have covered in year 12. So if you remember, transcription of the gene in the nucleus using RNA polymerase to convert DNA into mRNA. And then you have pre-mRNA strands that are spliced and introns are removed, so therefore leaving exons. And then mRNA leaves the nucleus via the nuclear pore, and then it binds to the ribosomes on the RER, and translation of the mRNA into a polypeptide through, for example, the joining together of amino acids brought together by tRNA. And then you need to know about anticodons and codons. So if you're not too sure on that, it might be worthwhile checking out one of my other videos on protein synthesis. So let's talk about regulation of transcription. Okay, if you didn't regulate transcription, then your cells would continually produce proteins, and you don't want that to happen all of the time. So as it says here, the gene that codes for the required protein is stimulated, and it's stimulated by something called a transcription factor, or a transcriptional factor. There are millions of these transcription factors, and each one has a DNA binding site that is specific to a certain gene. So when it binds to the correct region of DNA, that means that transcription will begin. So it produces mRNA, which then is translated into a protein. Now, the fact is transcription factors can turn genes on or off. So you could have repressors that can bind to the promoter region and prevent RNA polymerase from binding, therefore stopping transcription. But in your spec, they want you to know about, well, how are genes turned on by a specific type of transcription factor. In this case, it is estrogen. So we're going to look at how estrogen can switch on a gene and initiate transcription. So I'll just move myself out of the way. There you go. So let's talk about regulation of transcription with estrogen. Now, as you can see over here, we've got an estrogen molecule, and hopefully you'll already know that estrogen is a lipid, so it can easily cross through the phospholipid bilayer of the cell membrane via diffusion, and therefore it then enters the cytoplasm. You can see it entering the cytoplasm. Now, estrogen can bind to a receptor protein, so that receptor protein has a particular site which is complementary to the estrogen, and then it forms what we call a hormone receptor complex. So this complex is now known as a transcription factor. So as I just said, estrogen is complementary to the receptor molecules, and an inhibitor is released. Now, they don't show the inhibitor in this diagram, but it just means that the inhibitor is released, and therefore it means that it can then travel through into the nuclear pore, into the nucleus. So estrogen changes the shape of the DNA binding site, allowing it to then move through. So secondly, okay, so here we can see a shape of the DNA binding site is changed, okay, and it's the estrogen that causes the transcriptional factor to change shape. So it then travels through the nuclear pore. In the nucleus, the transcription factor, you can see it binds to a specific base sequence on a DNA promoter upstream of RNA polymerase, and then this binding stimulates RNA polymerase to start transcribing the gene, and it so stimulates protein synthesis, which you can see with the production of mRNA. So that's kind of how a gene may be switched on, but also we need to talk about how translation can be prevented using something called RNA interference, or interfering RNA. So RNAi molecules, they are small lengths of non-coding RNA, so they do not code for proteins. They regulate gene expression by affecting translation, so not transcription in this case, it's translation, okay, and they are double-stranded. So that means they're quite different to our other RNAs, and they stop out mRNA that's been transcribed from being translated. And the way that they do that is cut up the pieces of mRNA. So in your spec, they expect you to know about siRNA, which is in animals only, and also miRNA, which is known as microRNA in plants and animals. So siRNA is a short double-stranded RNA, as we just said, and it says one of the two siRNA strands binds with an enzyme. That enzyme is called RISC. Again, if you're doing AQA, you don't need to know that, it's just an enzyme. Then binds to mRNA molecules in the cytoplasm by complementary base pairing, and then this binding causes the RISC enzyme to cut the mRNA molecule in two, and it's cutting me in two, so I will move out the way, so it can no longer be translated into a polypeptide. So that's siRNA, and it's used in research to work out how particular genes work and understand how different proteins might relate to one another. So as it says here, so you can identify the role of genes in a biological pathway. You could block certain genes, so you could observe what effects occur. So this could then tell you what the role of the blocked gene is. Some diseases are genetic and can be caused by the expression of certain genes, so it may be possible to prevent the disease being caused by them. miRNA or microRNA, they are not fully complementary to the target mRNA, so they're much less specific. and you can target more than one type of mRNA. So they go through the processing stage in the cytoplasm to get to be a single-stranded, like siRNA, from a folded strand to a double strand, then two single strands, and then they're degraded with siRNA. What I will say is that when you look at an exam question talking about interfering RNA, it's pretty much the same process. They might talk about its use in, for example, HIV research or any other research. So it's quite applied, but you need to think about the basics of how these interfering RNAs actually work. So the next form of gene expression we're going to talk about is gene expression and cancer, which is the uncontrolled cell division. So we need to understand what happens in terms of the genes related to the cell cycle. So first of all, just to remind you about the cell cycle. So the cell cycle you would have learned about from when you talked about including mitosis, so interphase and mitosis, but cell division is tightly controlled by genes. Otherwise you would get cells being produced all of the time. So there are two particular genes we're interested in, and those are Proto-Oncogenes, and they stimulate cell division, and tumor suppressor genes that slow cell division. It's important to state that when new cells are being produced, there are certain checkpoints within which cells are checked, and if they don't, if they're not being formed in the correct way, they will be destroyed by a process of apoptosis. So let's talk about mutation of proto. So 3, 2, 1. Okay, so let's talk about proto-oncogenes. So as you can see by this diagram, this is when cells are dividing. So proto-oncogenes are important for helping the normal growth of cells. Okay, so when they mutate, they can become permanently activated. So the other diagram I've got there is the way that I teach my students to remember it is a bit like an accelerator in a car. So if you imagine that's foot to the floor, and therefore you're speeding up and speeding up and speeding up, the same as a mutated proto-oncogene producing lots and lots of cells, and that can lead to a cancerous growth. This type of mutation is known as then an oncogene. So the receptor on the cell surface membrane could be permanently activated. A growth factor is produced in excessive amounts. Next one is mutation of tumor suppressor genes. So again, tumor suppressor genes are they switch cell division off. So they normally help cells slow down, and they repair DNA mistakes or tell cells when to die. So almost like putting the brakes on in a car. So when tumor suppressor genes don't work properly or they mutate, then cells can grow out of control, which then can lead to cancer. So we need to talk a bit about mutagenic agents. So mutations can be caused by many different things, and it's in adults. It's related to changes to the base sequence of DNA. So things like ionizing radiation from UV rays or carcinogenic chemicals like those found in cigarette smoke or cosmetics or cleaning products. And some people can also inherit mutations in genes that make them more likely to suffer with some cancers. So for example, breast cancer. So tumours, you do need to know about the two different types of tumours. So first of all, we have a benign tumour. So the cells are not cancerous and they don't spread. They're slow growing. They do not metastasize. I can't even say that, metastasize. So that means that they don't spread, but they can put pressure on organs or cause blockages on things like blood vessels. The cells do have a normal shape. Whereas malignant tumours, well, the cells are considered cancerous and they start to spread to other tissues and organs. They are fast growing, they invade and destroy other tissues and they spread around the body, particularly in the lymphatic system, and they also have an abnormal shape. So we did mention estrogen and breast cancer, and we also mentioned about genes as well. So one of the things about estrogen is that some women are exposed to more estrogen than others. So for example, if you take birth control or HRT drugs that contain estrogen that help control menstruation or treat symptoms of the menopause, also starting menstruation early in life or starting the menopause later in life, and there is a greater risk of developing breast cancer. So that can also increase with age. Fat cells in the breast tend to produce greater and greater amounts of an enzyme promoting the production of estrogen. So the exact cause is still not fully understood. And I'm pretty sure if this is in an exam question, they will give you some data to analyze and evaluate. So on to looking at epigenetics. Now, epigenetics, I think, is the most exciting area of gene expression, because obviously when you are born, they say that you are born with the genes that you have, which you are, and the base sequences stay the same throughout your life. But increasingly, scientists are realizing that actually these genes through your life, through exposure to different things, to your lifestyle, can actually be switched on or switched off, and you can have genetic tags, and these genetic tags could be actually inherited by your offspring, by things that you do in your lifetime. So let's have a look at what we mean by epigenetics. So firstly, let's just talk briefly about the structure of DNA and the role of histones, because histones are very important in epigenetics. So chromatin is a double-stranded helical structure of DNA, okay, and that is wound around, complexed with histones to form nucleosomes, and they consist of histone proteins, because histone is a protein, and then they are folded up, and then they form loops, and then eventually they're compressed to make tight coiling of the fibers, producing the chromatid of a chromosome, and which is very important. And obviously if they're tightly coiled, then obviously the kind of mechanisms of protein synthesis can't go forward, so therefore genes can be inhibited, in other words not producing certain genes when the histones are not when they are tightly coiled. So first of all, what is epigenetics? Well, epigenetics here is an AQA definition. It's the heritable changes in gene function without changes to the base sequence of DNA, and I would learn that definition. So what I have here is a picture of a histone complex, and they're covered in chemicals or tags. So these tags are known as the epigenome, or epi comes from the Greek and means on, and these tags determine the shape of the histone, so the histone DNA complex actually, and they help to control the transcription of genes. So acetyl tags and methyl tags are very important. So let's talk about when genes are active. In other words, there's active protein synthesis going on, the genes are switched on, and that's known as acetylation. So when histones are acetylated, the chromatin is less condensed. So this means that the transcriptional machinery can access the DNA, allowing genes to be transcribed. Inactive, okay, so obviously if there's less acetylation, then they'd be inactive, but when you get more methylation, then they're also inactive. And as you can see by the diagram, that DNA cannot access inside the gene, okay, because the histones are tightly condensed or packed. So the addition of a methyl group added to cytosine bases of DNA, that attracts the proteins that then condense the DNA histone complex. So increased methylation changes the DNA structure so that transcriptional machinery can't interact with the gene. So the gene is not expressed. I hope you found that useful. So my next video will be looking at the recombinant DNA technology, and also I'll be going through some exam papers. So please do subscribe and make sure your bell alert reminder is on. I'll see you soon.